we can directly observe their molecular profile. The question is, how do we translate that into the ability to stratify patients? And in the context of a clinical trial, you can absolutely do that. And really what we have then is we observe a biomarker, in this case it would be a genomic type biomarker, defined treatment-specific subgroup, and then you can really translate that into a diagnostic so that when a patient comes into a doctor's office, a hospital setting, you could profile them and then accurately characterize their profile and segment them into whether they would respond to a specific treatment. So we really think this is the working model for the concept of patient stratification and is really part of the underpinnings of our analytics platform, and we'll talk a little bit more about that. So how do we actually do this? From a biomarker data perspective, it's not necessarily that we're always dealing with next-generation sequencing data, but as the case study today that we're going to walk through deals with that, I wanted to set the stage in terms of how that relates to our platform. So when we think about next-generation sequencing data and we think about the workflow, the breadth of knowledge that's applied is fairly extensive. And so on the left side, we have kind of what we call the data processing pipeline. We think of this as the primary or secondary analyses where we're doing alignment and calling of variants and annotation and those types of functions. Our platform is really geared towards where we have this red box, and that is more in the context of feature selection, so identifying relevant biomarkers. Then how do you take those biomarkers, especially if you have multiple biomarkers, which we often do, turn that into the ability to stratify patients. And then on the back end, how do we really visualize and report this type of information? We know that when we have complex data, especially in the context of clinical trials, being able to present and visualize information becomes extremely important. So this red box is really where we're going to be focusing on in terms of the demonstration today. So now just a brief introduction to what our platform looks like so that you have an idea of the underlying makeup of it as Toby's walking through the demonstration. First, just want to speak to what the motivation of the platform was. I think we all know that not just the pharmaceutical industry, but maybe more broadly speaking, the healthcare industry is moving towards a data-driven approach. It's also heavily rooted in being multidisciplinary. So that's what the figure on the right is really trying to demonstrate, that a data-driven personalized medicine approach exists at the intersection of multiple disciplines. Well, we happen to be from the analytics perspective. There also has to be heavy input from a clinical perspective, regulatory, market access, and your pharmacogenomic and translational research team. So that is really the underpinnings of how and why we built the platform the way we did. We understood where research existed, and we engineered a platform from the ground up to address the needs of these types of efforts. More specifically, the platform was designed to do a few key things, and I'll walk through those very briefly. The first one being data integration. And what we mean by data integration here may be a little bit different than how you normally think of data integration. We're not thinking of integrating necessarily different types of databases into a much bigger repository, but rather we're thinking of integrating different types of biomarker data together so that we can simultaneously look at things like, say, somatic mutations in gene expression. And we'll go through how that looks today and actually what the added value potentially is of overlaying different types of biomarker data together simultaneously in the same platform, in the same model. This can then translate to biomarker discovery, so the concept of feature selection, really understanding the relevant genomic characteristics that are driving treatment response, and then ultimately translating that into patient stratification, which is really what the platform was designed to ultimately do, stratify patient populations for the purposes of ultimately developing in vitro diagnostics such as LDTs, et cetera. The platform was developed so that it's multimodal in nature, and we say multimodal because there are really two components that drive the platform. The first component being an advanced Bayesian analytics underlying algorithm, if you will, that can take genomic data and transform it into these molecularly defined patient populations that you can then ultimately use to stratify and segment populations, for whether it's for developing diagnostics, for developing more efficient designs of future trials. That was the initial driver of the platform.